Buenas tardes. Buenas tardes. El día de hoy no tenemos asuntos generales, entonces me pidió nuestra directora y secretario general que vayamos directo a la introducción del ponente de hoy. Y en español quiero dar dos indicaciones. Una de ellas es que tenemos unos micrófonos al lado derecho e izquierdo para quienes quieran hacer preguntas. Por favor, se levanten y hagan fila para las preguntas. Una razón es importante para esto es el orden y la otra porque se está transmitiendo por YouTube. La otra situación es que si alguno no puede hacer la pregunta en inglés, estamos dispuestos a intentar hacer la traducción de esa pregunta, entonces la pueden hacer en español. Esto va dirigido especialmente a los estudiantes para que se animen a preguntar. So, good afternoon. It's a pleasure for me to introduce Rob Sears. He's a research assistant professor in NYU in the CNS, in the Center for Neural Science. And he also works as a research scientist in the Emotional Brain Institute in Nathan Kline Institute. And he works studying the hypothalamic feeding peptides and their influence on the nucleus accumbens in the brain when he was a graduate student. And he did this in studying a major in biology and philosophy in Henry's College in Texas. And then he went to do the PhD in Yale University. And then he did a postdoctoral position, in New York University with Joe Ledoux. A lot of you know his work, which is about emotional and fear, learning, and memory. And now he has been for a long time publishing articles, and his articles has been published in journals such as Nature, Neuron, Cells, PNAS, Learning and Memory, Neuropharmacology, Journal of Neuroscience, +1, Cell, and he's in the editorial board of scientific reports, and he's a reviewer editor of different journals. I'm happy to say that he has students for PhD and also postdoctoral students. And one of our students from this institute that she did her major thesis, master thesis in the Maestría of Neurobiología and the PhD in the program of Ciencias Biomédicas, which is Chris Siller, and was my student. She's now working with him with a postdoctoral position. So it's very nice to know that our students in the institute can go to other places and work with people like Rob Sears. You will see his work, and I hope you can enjoy his talk. And he has, Rob has this big net of people that work in emotional brain problems. And this is very good because it's not only, he not only produced papers by the results of his laboratory. They do this big connection with Joe Ledoux and do all this research, which I think is amazing to have that, Rob, to have a big net that also includes people from other countries and not only in the United States. And well, I think that your work will be talk of you. So thank you so much for accept to be here, and thank you so much for being presential, because I know that has been difficult and traveling is not easy now. So I hope you enjoy your stay here. Thank you.
Thank you so much, Gina. I am more than happy to be here, and I really want to thank the grad students from the bottom of my heart. Thank you so much for getting me away from my desk and staring at the computer screen and giving talks sitting down. This will be my first talk I think I've given in almost 2 years standing up, so I hope I can remember how to do it. So today I'm going to be talking about survival circuits in the brain when it comes to surviving threats in our environment, and some of the strategies we use to evade those threats or avoid them. So I think we can all agree that the world is a somewhat dangerous place. The question is, however, how we cope with these threats so that we can remain engaged in the world and live happy and healthy lives. Can you guys see the arrow? No. How do we do the, can you help me with the pointer?
Okay. Next.
I just need the laser pointer.
Okay.
Okay. Okay. Okay. Anyway, so at the top you can see some of our, some of the threats we've been dealing with in the world. And below you can see some of the ways that we deal with those threats by taking proactive steps. For example, wearing a mask, washing your hands, getting vaccinated, going out and voting in case you don't like what your government's doing. These are examples of adaptive coping behaviors. However, on the right you can see other more maladaptive behaviors, which are just kind of curling up in a ball and staying at home and avoiding the world, or turning to drugs of abuse or alcohol. So I'm sure everyone's familiar with the tragedy that occurred not too far from here recently, the Metro bridge collapse. I just want to use it as an example of what we study. So imagine you're a pedestrian on the ground and you hear a loud, thunderous noise, as this survivor calls it. So at some point in your life, you learned that loud, thunderous noises probably mean danger, right? So what do? Well, the first thing you probably do, the automatic, unconscious thing you do, is you freeze. Most animals, most vertebrates do that when they encounter a threat. The next thing you can do is turn around and book it out of there, try to avoid the threat. However, if you're a trained medical technician or maybe a combat veteran, you may be inclined to run towards the danger to see if you can help people out. So those are ways that you can cope with the situation while it's happening. People who've experienced this, it's traumatizing. They may go home and suffer from some post-traumatic stress for a while, but then, most people get over it and they get better. However, some people don't. A minority of people, but some people don't. So that's what we're interested in, is how people cope with threats in the situation, but also what comes after, how they recover. So how do we learn about the world, and how do we adapt to our environment based on what we've learned? So the first way is through classical Pavlovian conditioning, and we do this all the time. This is, imagine there's sounds, visual cues, smells, and they're biologically meaningless until they're paired with something that's biologically relevant. For example, a shock, that's biologically relevant. You can feel that. So the tone, the sounds, the smells, the sights, those become conditioned and connected to the unconditioned stimulus. Those are conditioned stimuli. Another way that we learn is to, once we've made these associations, to use that information to do something about threats that may be occurring or about to occur. And this is known as instrumental or Pavlovian conditioning. So this is you can see a rat's pressing a lever in order to avoid a shock. Now, early in our evolutionary history, with the amniotes and even before, we developed brain circuitry that's critical for these survival behaviors, these so-called survival circuits. And they're homologous between all these species. The circuitry is very similar. And at the center of it all is a structure known as the amygdala, the temporal lobe. And on the left here, you can see the human amygdala, and then on the right, you can see the rat amygdala. Very, very similar structures, hooked up very similarly in both the rat and the human. The amygdala is critical for taking sensory information that may be irrelevant and connecting it to information that is relevant to the animal. So the amygdala is not one homologous structure. It's actually split up into a bunch of structures, and a few of which I'm showing here. We have the lateral amygdala, the basal amygdala, and the central amygdala. There are also other nuclei, the medial amygdala, the cortical amygdala, and you can even split them up further if you want, based on cytoarchitectonics and circuitry. So we know from decades of research that the lateral amygdala is kind of essential for the functioning of the amygdala, for learning. So sensory information makes its way there, the conditioned stimulus, the unconditioned stimulus, and through a Hebbian process, connections are made and plasticity occurs. And then what happens is the LA then communicates with, for example, the central amygdala, which is a major output of the amygdala, and that is critical for that unconscious freezing response I mentioned. It also projects out to autonomic centers to get your heart rate going and your breathing escalates. I will also mention projections out from the basal amygdala are important for more proactive behaviors, and I'm not going to really talk about our work on that, but just suffice it to say the amygdala instructs the ventral striatum to, which is kind of a motor limbic interface, which is important for eliciting avoidance responses, for example. So to study classical or Pavlovian conditioning, we use this procedure in the lab in which we pair usually a tone or a sound with a foot shock, so in close temporal proximity. After the first pairing with the second presentation of the tone, you can see the animal has already started freezing, so they learn very, very fast. You can then take that animal and put them in a completely different context that looks and smells different. The animal usually is exploring, but then when you play the conditioned stimulus again, or what has become the conditioned stimulus, the tone, the animal will freeze. And this lasts for the lifetime of the animal. People have tested years later, or as long as a rat lives, and you can play that tone and the animal will still freeze. So this is very strong memory, very quickly formed, very much hardwired, the reaction, the freezing reaction. So here's an example of a rat that's been conditioned. So he's exploring the new context, and then you'll see at the bottom it says the tone comes on, and then you'll also see a light come on. And when that happens, the animal begins to emit this species-specific defense response, which is freezing. Again, it's like a reflex, right? So we became interested in plasticity in the LA and the neuromodulatory mechanisms that are happening there in order for these memories to form. So we looked in the amygdala and we found that there are a lot of axons, noradrenergic or norepinephrine expressing axons. You can see they're very strong in the LA. So before I joined the Ledoux lab as a postdoc, a postdoc at the time, Dave Bush, showed that if he infused the beta adrenergic receptor antagonist propranolol, which blocks noradrenergic activity in the LA, if he infused it prior to training, he saw that animals, their freezing reactions when tested later were blunted. So he could test them 3 hours later, that's short-term memory, blunted. Long-term memory, 24, 48 hours later, blunted. So it appeared they couldn't learn. To ensure that it wasn't some sort of consolidation process, which occurs after the acquisition process, he infused the drug after training and found no effect whatsoever. So this suggests that norepinephrine acts through beta receptors in the lateral amygdala for the acquisition of the emotional memory. And consistent with this, working with Hilary Schiff, grad student in the lab, we showed that activating beta receptors actually enhanced memory when you looked at it after 3 hours or longer. So the major source of norepinephrine in the brain is the locus ceruleus, which is back in the brainstem. In rats, it's about 2,000 cells. And we're looking at what's expressed in the locus ceruleus. One of the most highly expressed, if not the most highly expressed receptor in the locus ceruleus, as you can see down here in the bottom left corner, is the Erection 1 receptor. So the Erection system is localized to the lateral and perifornical hypothalamus. The cells are exclusively found there. As you may or may not know, the hypothalamus is kind of like a command center for the brain. So it makes sure that we're always doing what we prefer to be doing, that we're not hungry, that we're not thirsty, that we're not threatened. So it's always trying to get us back to homeostasis or allostasis. So the Erection system has its fingers in a lot of pies, so to speak. It does a lot of things. It's important for wakefulness, for arousal. It's actually been shown that the Erection system can act to lead to waking through the locus ceruleus, for example. It's important for maintaining wakefulness. And here's where I mention an interesting fact, and that's that humans or animals without Erection or Erection receptors they're narcoleptic. So they cannot maintain wakefulness. But also some are cataplectic, meaning when something either scares them or excites them suddenly, they lose all muscle tone. And it's interesting because they lose consciousness, but not all muscle tone. So what this told me when I was thinking about Erection system's involvement in threat learning was that it's important for transitioning from, a kind of a baseline state to a state of I need to engage, right? And I need to respond to either a threat or something that I want to approach, like food. Obviously, it's important for food consumption. I just mentioned food. And it's also important for reinforcing behaviors. So it's been shown mainly to be important for reinforcing food-seeking behaviors as well as drugs of abuse. So the first thing, I hypothesized based on our noradrenergic data in the LA and Erexion's connection to the LC that Erection was actually modulating LC, which was then going on to modulate the LA. So the first thing I wanted to test was, does Erection activity in the locus ceruleus control threat conditioning? So these experiments are very similar to what you saw with David Bush and Hilary Schiff's experiments, except I infused the Erection receptor antagonist directly into the locus ceruleus. And the results were very similar. I found on the left here that giving the drug, a higher dose of the drug, prior to training, their freezing at long-term memory points 24 hours later was severely reduced. And if you hop over to the far side, you see that pre-training short-term memory, which is when I test 3 hours after training, is also reduced. So, and then when you take in the middle the post-training infusion of the drug, there's no effect whatsoever. So this looks a lot like those noradrenergic propranolol experiments where the animals do not seem to acquire the memory. So next, I turned to optogenetics to nail down that the connection from the Erection system to the locus ceruleus was indeed important for acquisition. And I kind of assumed everyone knew what optogenetics is, but briefly, it's you can express channels that respond to laser light in cells of your choosing. You shine a laser on it, and you can either inhibit the cells or you can activate the cells. Or you can also activate or inhibit the axon projections to target regions, which is pretty cool. So I got this virus that was specific to Erection cells from Luis de Lucia's lab. It was a lentivirus and worked very well. Here I'm showing that I just wanted to make sure it was activating the LC, so I just did a quick Cfos experiment and found that only when in animals expressing channel rhodopsin and the axon terminals was there an increase in Cfos, and not in animals that were expressing M-cherry and M-cherry control, so they weren't expressing excitatory opsin. So when I stimulated during the unconditioned stimulus, actually, during training, and then tested the animals later, I found that stimulating Erection terminals and LC led to an enhancement in the freezing behavior, the conditioned response. Whereas we didn't see that in control animals that got the laser light or channel rhodopsin expressing animals that didn't get light. Now, Erection cells have been reported to also express things like glutamate. So we wanted to make sure that this was due not to glutamate, but to Erection signaling specifically. So we infused the Erection receptor antagonist into the LC and then did our laser stimulation and found that on the bottom right that there was a reduction in freezing. So indeed, it seems that Erection acts in the central amygdala, or in the LC, sorry, to drive the acquisition of conditioned freezing. So the next thing we wanted to test was whether or not the LC is projecting to the basal amygdala as well, or I'm sorry, the lateral amygdala, in order to drive acquisition. And bear with me, this is a little complicated. This is what's called a disconnection experiment. So it's a bilateral circuit between the hypothalamus, the LC, and the LA. So one side, if you take it out, the other side will compensate, and learning should be intact. So what I did is I infused the Erection receptor antagonist on one LC, and on the same side, I infused propranolol into the LA, which left the other side intact, and animals learned just fine. However, in B there, if you take a look, what I did is I took out the Erection system, the Erection activity in the LC on one side with the antagonist, and then on the other side took out the circuit from the LC to the LA with propranolol. And taking it out at different nodes on each side reduced the acquisition of the conditioned response. So this does suggest it's a serial circuit between Erection cells, LC, and LA. So we also noted that the norepinephrine fibers were also in the central amygdala. And the central amygdala, as I mentioned, is important for the conditioned response, the actual emission of the freezing behavior. So we wanted to test whether or not central amygdala, norepinephrine in the central amygdala modulated freezing behavior. And to do this, we would have to do our manipulations prior to the long-term memory test or the expression test. And Joy Gu, a very talented grad student, first infused propranolol prior to the expression test. And it was a pretty modest effect, but we did see a reduction in freezing, the expression of conditioned freezing response. And then the next day when we tested them, when the drug had washed out, there was no difference, suggesting that there was no kind of long-term plastic effect from these manipulations. She also infused the agonist, procaterol, and found an enhancement in freezing. which again washed out the next day, and they were comparable in their freezing levels. So next, Joy turned to designer receptors, DREAD, designer receptors exclusively activated by designer drugs. So like with optogenetics, you can target cells with a virus that expresses a receptor that's been engineered from the human muscarinic receptors. But it's been engineered so that it doesn't respond to acetylcholine anymore. It only responds to an exogenous ligand. Initially, clozapine and oxide, but they've since made other ligands that can activate it. So these ligands shouldn't do anything else to the animal. They should only target the receptors that you've expressed. And then we have two kinds of receptors, HM40I, which can inhibit cells, or it actually hyperpolarizes the cells and decreases their chance of firing. Or you can use HM3DQ receptor, which depolarizes the cells and increases their chances of firing. So to do this, we used a norepinephrine cell targeting promoter virus that was generated by Elena Vasey in Gary Aston Jones's lab, a very talented postdoc who now has her lab at UMass Amherst. So this is just showing on the left that DBH is an enzyme found in noradrenergic cells. In the middle is the HA tag, which is on all DREAD receptors. And then on the right, you can see the overlay where there's a perfect match. So very beautiful, very specific. And now I'm going to brag and show you that we got the cover of Molecular Psychiatry. I honestly argued with the editor. I was like, I think this is a prettier picture, but they went with this one, so whatever. We got the cover, it was cool. So we have 3 different flavors of DREADs. One is expressing HM3DQ, one is expressing HM40I, and the third is expressing the fluorophore M cherry, which does nothing. It's just a marker, fluorescent marker. So we inject animals with one of these. Then we train them and infuse or give them IP injections of CNO, the ligand for DREAD receptors, prior to the expression test. So the first receptor we tested was the excitatory receptor. And you can see the data at the top left, BL is baseline, so that's their freezing before anything. I was happy to see that they weren't freezing at baseline, that it was not just non-specific freezing by activating the locus ceruleas. And in the middle, you can see those are M cherry controls that got CNO. They're freezing fairly well. But when you activate these neurons, they freeze quite significantly more. And then the bottom right is just showing that they pretty much come back down to normal the next day. So again, there's no long-term plastic effects of these manipulations. Next, we use the inhibitory DREAD. The effect, as you can see in the top left, was more modest, but it was significant. And then on the right, you can see that the effects kind of wore off the next day as well. Now, when we looked at these brains, we noted that in the central amygdala, we could actually see the DREAD receptors. And this is not surprising because these muscarinic receptors, the endogenous human muscarinic receptors, are known to traffic to axon terminals and to actually excite or shunt axon terminal activity. So we decided to target it directly by doing our injections in the LC and then cannulating the central amygdala and infusing CNO directly into the central amygdala to confirm that that's the circuit we're working with here. So on the left is just the control M cherry with CNO. And you can see the animals are freezing at a modest rate. They also got propranolol, I should say. HM3DQ is in the middle. You can see a nice, robust freezing response. However, if we infused propranolol in the central amygdala and then CNO, it significantly knocked down the effect of activating the LC to central amygdala circuit. So this suggests also that LC projects to central amygdala and activates beta receptors, and which is important for the condition freezing response. And again, targeting the axons in the central amygdala With CNO, we get a nice reduction in the freezing response, which the next day comes back to normal. So to summarize this part, this is all really the Pavlovian conditioning studies. Erection can activate LC, which then projects to the lateral amygdala, and through beta receptors, positively modulates the formation of memory, of fear conditioning memory. We also showed that the LC projections to the central amygdala through beta receptors is important for eliciting the condition response. All right, so I spoke to you about some of our work on classical or Pavlovian conditioning. Now I'm going to talk to you a little bit about our work on instrumental or operant conditioning. Now, around the same time Pavlov was doing his thing with dogs, salivatory reflex, this other guy, Vladimir Bekhterev, was working at the same time, and he was highly respected. And he was using a procedure known as signal active avoidance, or at least we call it that now. So it involved learning a Pavlovian association between some sort of stimulus and then a shock. And a lot of this work was done in people. However, the difference between his work and Pavlov's work is that people were given the option to escape the conditioned stimulus. So when the conditioned stimulus played, they could remove their hand and not get shocked, and completely avoid the unconditioned stimulus. So since then, this has been adapted to animal models. And I'm just going to show you an example on the top of an animal that successfully avoids, is a good avoider. So now the tone is on, crosses over casually, tone's off, doesn't get shocked. Guy on the bottom can't overcome his freezing response. He's just rigid with the freezing reaction. He gets shocked and escapes the shock. So when he crosses over, the shock turns off, and so does the tone. So I just want to mention as an aside that the animals that freeze the whole time, that don't learn the task, are a small percentage of all the animals. And we like this as kind of a model for taking information, learning about our environment, and coping strategies, proactive coping strategies in order to deal with threats as they come. Chris Kane, who's my colleague, showed that if you lesioned the central amygdala and took away that major output for the freezing reaction, animals will immediately start avoiding, which suggests that these animals actually know they can avoid, but they just can't overcome that freezing reaction. So we're very interested in that response competition. It's one of the major focuses of our group. So back to Erection. I can't escape it. So I was very inspired by this perspective piece by Gary Aston Jones and his colleagues, in which he suggests that Erection is completely valence nonspecific. It's critical for motivating action in the face of need. So basically the suggestion is Erection coordinates and drives actions to achieve homeostasis or allostasis, as I mentioned before. Now, in the lab, we developed a vector, an AAV vector, which is nice, to express an inhibitory opsin, Arch 3.0. You can see on the left, that's the expression. Those are all the cells and axons coming off. And then below, you can see perfect co-labeling between the virus expressing Arch and Erection cells. And when we look at targets of our cells, we're not surprised to find that it projects to the central amygdala, which has been noted, specifically the central medial amygdala. But it also projects to the ventral tegmental area. And it's been known, based on a lot of appetitive research on the Erection system, that Erection does act in the VTA. It activates these neurons and seems to be important for conditioned appetitive learning, or learning and expression. The VTA is part of the mesolimbic dopamine system, which is composed of the VTA and the ventral striatum. And this system is essential for reinforcement learning. So, given that signal-active avoidance we think is an instrumental behavior and requires some sort of reinforcement, it made sense that the Erection system may be modulating the VTA for learning of the reinforcement behavior. So our hypotheses are that Erection acts in the central amygdala to actually inhibit central amygdala output, inhibit freezing, thus permitting the avoidance response. And this was based on kind of a paucity of data, electrophysiology data, showing that Erection tends to inhibit cells in the central amygdala. So we kind of extrapolated that it's actually inhibiting the output of the central amygdala. Whereas action in the VTA is important for invigoration or reinforcement of the avoidance response. So so I want to actually say that a lot of this data is, I'm going to call it preliminary. These are long experiments, and we're trying to add a lot of animals to it. We're doing males and females, so right now we're currently adding more and more animals to add more power to the results, but I still think it's pretty exciting. Gina mentioned that Chris Siller works in my lab. She's a very talented postdoc, and if she's any representation of the students from this university, I have to say I'm incredibly impressed. She's been great to work with, really amazing. Anyway, enough gushing. So Chris injected the virus into the hypothalamus to infect Erection neurons, and then she has to let the animals recover for a while, because it takes a while for these opsins to get to the axon terminals. And then she implants fiber optic cannula bilaterally, either in the central amygdala, it's either on the top right, it's kind of hard to see the tracks, or the ventral tegmental area. So after recovery, she then hooks the animals up to the tether, which is hooked up to a laser, and habituates the animals to having a tether on their head. And she just lets them explore the boxes for a little bit. She then trains the animals with the tether. On the bottom left, you can see for the expression test, she trains the animals with no laser on, because we're going to train them up, get them to learn the behavior, and then we're going to manipulate. On the right is an example of an acquisition test where manipulations start right at the beginning to see if the Erection system in different targets is important for actually acquiring behavior. And then at the bottom, I'm showing you that we test them in extinction. So we take away the shock, and then we test what happens with the manipulations, what the animals do. So the first thing we looked at was the central amygdala circuit. And contrary to what we expected, so I'm showing the data here in latency, which is a kind of a proxy for avoidance responses sometimes. If it reduces the vigor, the tendency of the animal to do it, likely is it's affecting the avoidance response. So you can see on the left here that these are the same animals. These are within subject experiments, counterbalanced. And it seems like whenever the circuit is shut down from the Erection cells to the central amygdala, that there's a reduction in the latency for the animals to avoid. And you can see that the data broken down on the right by blocks of 5 for 15 CSs. And you can see that even though these laser off animals are extinguishing, their latency is getting longer and longer and longer because they're realizing it's not being reinforced with a shock. Laser animals just keep going and keep going fast. So this is in contrast. So it suggests basically that Erection, when it's normally active in the central amygdala, is important for maybe the extinction behavior. Okay, so next we started to look at the VTA circuit, and so this is also during expression. As we expected, we saw an increase in the latency in these animals when we shut down Erection activity in the VTA, and that's on the left. And then on the right is just, we were like, let's see what happens. Let's see if they ever recover. Let's see if they ever can, their responses, latency comes down. And it just never did. She kept putting them in. So these animals really just, they were just not nearly as good without orexin, VTA. So next, we wanted to see if the orexin to VTA circuit is important for the acquisition of the behavior. And this makes a lot of sense if VTA and the mesolimbic dopamine system is important for reinforcement learning. So inhibiting the circuit definitely, now I'm showing avoidance responses on the left, definitely reduce their ability to learn the avoidance response. On the right, you can see that their latency was delayed. Now, although we don't have anymore the excitatory opsin version of this vector, we decided to just go ahead and inject the hypothalamus with a generic channel rhodopsin, actually cheetah virus, and stimulate the terminals in the VTA. And Chris found that this enhanced the acquisition, as you can see on the left, from day one. And the latencies were way reduced. So these animals were much better and faster at the avoidance response. So to summarize this, the Erection system seems to modulate the central amygdala to drive freezing, maybe, because when we take it out, they're better at avoidance. We have to follow up on that, obviously. And then also it acts on the VTA to modulate avoidance learning, to promote it, and also the expression of the behavior. Now, if I put all this work together, you can see that well, on the left is the Pavlovian stuff, and on the right is the instrumental stuff. So what's at the top? Well, it's the Erection system. So that's why I like to call the Erection system kind of a master modulator of survival circuits, because you can see it's projecting down to other catecholinergic and memory nodes in the brain, and seems to be really important and really influence these targets. Now, that's not to say that norepinephrine isn't important and that we're not still studying that circuit. We're actually now, many of you may be familiar with all this brilliant work that started with Luthi and David Anderson on the microcircuitry within the central amygdala. And so now we're actually looking at noradrenergic activity in the central amygdala on a microcircuit scale, and how that may tip, act as kind of like a rheostat to drive the animal to freeze or to get the animal to avoid or even escape. So that's what we're doing now. So with that, I'd like to thank a lot of people. I'd like to thank Erica Andrade, who is a molecular biologist who made our vectors and viruses. Gary Aston Jones, and at the bottom, Elena Vasey, who we collaborated with on using the PRSXA, the LC-specific virus. Chris Kane, my colleague, who's a brilliant behaviorist, and we couldn't do any of this behavior without him, without his input. Our techs, Lindsay Laughlin, Eric Martinez, Daniel Maloney, they've all gone on. Lindsay's at UCLA, Danielle's in a marine biology lab, and Eric Martinez is in medical school. Shauna Samuels, she's also in graduate school now. Those are some amazing technicians, got very lucky. And of course, last but not least, Chris Siller-Perez, who has just been amazing for the lab, and she's going to continue working hard, and we're going to hopefully get this story out pretty soon. So, and I actually threw this up here. I just wanted to mention something we think is happening. So, Michael Fanslow has this idea that there's a predatory imminence scale or continuum, in which, you know, if you're right in the thick of a predator attacking you, you're at the super high end of the survival circuit activation or the amygdala activation. However, as you move down the scale and threat's a little less imminent, there may be signals that there could be a threat nearby. That's where probably avoidance happens. So Bowles argued that he thought that all avoidance was, Robert Bowles argued that all avoidance was probably fight or flight, species-specific defensive responses. But we believe that, everybody learns the avoidance response, all organisms learn it. And this is occurring kind of farther down on that scale of threat imminence. So, anyway, so that's it. Any questions?
Hi, thank you very much for the talk. I have two questions. The first is regarding the activity of the locus ceruleus, the orexinergic neurons during freezing. Do we know what those neurons are doing?
What the locus ceruleus neurons are doing during freezing?
I'm sorry, the orexinergic neurons. Are they firing? What is the activity?
During freezing? Yeah.
Yeah.
So they, that's a good question. I mean, we do know that they fire during conditioned stimuli. We know they fire during unconditioned stimuli. During the freezing response, I'm not sure if they're still going, to be honest with you.
Because
So
I
don't know
imagine
if
it would be important to know what they're doing because they are the master modulator, right? They will engage the amygdala.
Absolutely.
So hence my second question is like, who's before those neurons? Because I had the idea that the master modulator for freezing
right.
and all this, it was the amygdala, but now you're proposing is the orexinergic system. So who would be before?
What's prior? So that's a good question. I mean, it receives all kinds of input. Orexin neurons receive all kinds of inputs from all over the place, including reciprocal connections from LC, from central amygdala, from VTA, from RAFA. So these are actually, so it's also talking to those things, those nuclei. But I do know that orexin cells are activated by salient stimuli, loud noises, shocks, food, palatable food. Everything that's been checked that's salient activates orexin neurons.
Okay, thank you very much.
Thank you.
Well, congratulations on this great talk. I have a question. You have talked about conditioned fear, Pavlovian type, and then avoidance, operant type of learning. Now, for operant type, these avoidance responses have to do with the activity of the striatum, amygdala, and hippocampus, whereas Pavlovian conditioning has more to do with hippocampal and amygdalar activity.
Right.
So how did you, how could you explain that the operant type of conditioning has these three structures involved, whereas the classical type of conditioning only has to do with hippocampus and amygdala?
Right. Well, so I will say that avoidance, signal-active avoidance, is actually, it's two factors. It requires two factors. It actually requires Pavlovian conditioning, right? So you first need to make associations. So you need your amygdala, you need your hippocampus, but avoidance does, it takes, is the next step. So you use that information that you've, those associations you've made to instruct the adaptive behavior. And that involves, I think I mentioned earlier, outputs from the basal amygdala. So that information is transferred to the striatum, the ventral striatum, as well as the dorsomedial striatum. I didn't show you any of the data yet, but we do know it projects to the accumbens for the expression of the behavior, and we have data to suggest it's also dorsomedial striatum, which is not surprising. And we think that probably, ultimately, when it's overtrained, just like anything else, that repress any other operant behavior, that it's going to require the dorsolateral striatum. But the first thing that happens is the Pavlovian association, and then you layer on top of that when the animal has the option for the instrumental response that they can learn it. They actually kind of stumble on the response accidentally, and once they're like, oh, this works, this avoidance, this action works, they immediately start doing it.
Okay, thank you. Second question. A long time ago, Dick Thompson postulated that the cerebellum was a main structure to produce classical conditioning.
That's been shown a lot.
In this paradigm, do you think that the cerebellum has something to do?
In active avoidance, possibly, but I mean, it's probably, most of what it's doing is probably Pavlovian, in the Pavlovian conditioning. But I'm not, yeah, maybe. I could only speculate.
Okay, thank you.
Sure.
Thank you for your seminar. Regarding this circuit, hypothalamus, locus ceruleus, amygdala, is there a circadian modulation of this circuit?
I'm sure there is, yeah. I mean, circadian circuits hook up to and they modulate LC, right? And Erection, we know, can modulate the circadian circuit directly. So, yes, I'm sure it's involved.
Thank you.
Any more questions?
I have a pretty little questions, and it's just one. And it is, what is the relationship between fearing and eating behavior, and is it correlated with orexin?
I'm sorry, what was the first? Fear in feeding?
Yes, fear and eating.
Oh, fear and eating.
Behaviors.
Yeah, so I mean, they're in competition with one another, right? I think our default mode, so that's another survival circuit, as Joe Ledoux likes to call it, right? So you have your circuitry, or it's not one circuit, but you have your circuitry that's important for feeding and your circuitry that's important for staying alive. We're actually getting into some questions, doing some work to look at, survival from a threat always overpowers your need for food, right? And if you look at an animal foraging, they have a level of anxiety always, right? And they have to be aware of their environment and that there are no threats. But as soon as any sort of indication of a threat is there, then they'll book it back. So, but we know the amygdala, for example, is important for learning appetitive behaviors as well as aversive behaviors, and exactly how, if that's based on topography and how it hooks up to other brain regions, is not completely fleshed out yet. But a lot of neuroscientists are interested in that question.
Okay, thank you.
Thank you for your talk. My question goes, well, towards drug abuse. In the last slide you presented, where would it fall in the response, and how would you integrate it into the circuits you described? Because, well, drug abuse is more of an active response more than freezing, but it's
right.
Maladaptive. What would you have to say about this?
Oh, I see what you're saying, yeah. Well, drugs of abuse have a pharmacological effect, and that effect is on dopamine neurons, right, and on dopamine release usually. So what it's doing, if Erection is activating the VTA neurons in the mesolimbic dopamine system, and those dopamine cells are supercharged to release dopamine. Erection is going to be depolarizing like normal, and then it's just going to be, it's basically going to stamp in and reinforce that behavior a lot stronger. So when the drugs are on board. But you know, the whole VTA to Cummins thing, I don't think it always has to be, it doesn't necessarily have to have a valence. It doesn't have to be, you know, approaching something or receiving something that you like versus something that you don't like. It's just it's kind of a mindless reinforcement circuit, right? The things that we like and the things we don't like, we now are learning have more to do with, I don't know if you're familiar with Craig Barrage's work, Kent Barrage's work, looking at like mu-opioid receptors and stuff like that. That's more the liking versus the wanting. The wanting is more the mesolimbic dopamine circuit, just kind of reinforcing, I'm doing this behavior, I don't really know why, right? So that's kind of how I see them working together. Does that make sense?
Yeah.
Okay,
Well,
cool.
yes, thank you.
Sure.
Thank you very much for your seminar. It's very interesting how orexins are regulating many different processes in the brain. So have you explored the possibility of differential stimulation on those orexin neurons to affect your results? Or even if hormones are affecting your results? Because I wonder how orexin neurons know how to regulate arousal or stress response or feeding or learning or drug addiction. So they are still involved in this, or could be involved in this, in addiction, influencing BTA.
Yes.
So,
I mean, I think stress levels absolutely will lead to a different outcome in terms of orexins, how it acts on different brain targets for different behaviors. So some of the earliest work with orexins was Louis Delisle that showed that orexin and CRF actually work together for the stress response. So you can't have one without the other for stress. That research kind of fell away. I haven't seen much on it in quite a while. It's not the main focus of his lab anymore, or other labs. But I definitely think there's, depending on the baseline stress of the animal, it's going to change how it targets or influences other parts of the brain. A recent study by Luis's lab showed that in the BNST, orexin can target different cells, and you can actually have appetitive versus aversive kinds of responses, depending on which cell subtypes it is. So, although it's very confusing because other people like Denis Berdikoff have shown that they just cannot find a topographical explanation for these different appetitive aversive functions. It seems like all the cells are that go to the VTA all go to the VTA, and they branch off and go to the other regions too. So it seems to be pretty, it can't be explained by how they're hooked up, you know what I mean? So there's a lot to be figured out.
Hello, sir. Thanks for your research and for your talk. Sorry. Do you believe that with your research and seeing all this orexin or peripherin, all the circuits involved in freezing or fleeing or avoidance, do you believe this basic research could evolve to how do I say it, like a staple to some translational research to be made? And what do you believe it could be used for in clinical translational research?
Yeah, so I'll go on my spiel now for a little bit, and that's that flooding or extinction therapies, which is what is done for people with phobias or post-traumatic stress, has very limited efficacy. So what an example is, let's say you're afraid of spiders, the therapist will get you closer and closer to a spider, right? And then eventually have you handle the spider. And the idea is to flood you with the stimuli for you to realize it's not a threat, right? However, when you go home or go on vacation, you're not with your therapist, and you encounter a spider, it's all going to come flooding back, right? You're going to that successful extinction that you had in the clinic is all of a sudden poof, right? So what we like about the active avoidance behavior is the fact that these animals have a sense of self-efficacy. I mean, I'm anthropomorphizing them, but they are in control of their situation. And in people with anxiety, stress, depression, that's a hallmark of those diseases. You feel like you have zero control over your life, right? So if you can use a training procedure to teach people you are in control, and maybe give them, some sort of help with, I don't know, an Erection drug or something like that, then I could see maybe some progress. Because if you learn internally that you're in control, that I have agency, it doesn't matter where you are. That won't be contextually specific like extinction training is, right? It'll be something you carry with you. So this is kind of what we're plugging, and that's the translational angle.
Okay, thank you very much.
Sure.
So is the Erection system also involved in extinction?
Yeah, there's been some evidence that it is, that it actually is pro-extinction in mice. I don't know. I did expression tests, and I never saw an effect in rats with my manipulations on expression, which I was surprised by. But people have shown expression in mice effects and extinction effects. So could be a mouse or rat versus rat thing.
Hi, I have a really easy question. In your last slides, you have grooming and another word that I can't understand.
I have what?
Two words in your scale of anxiety. Another one.
This one?
No, when you have a scale.
Oh, I'm sorry. Yeah. Yes, predatory imminence.
Yeah, that's one. Grooming and another one, I can't understand that.
You can't understand that. Yeah. Okay, so let's say okay, actually let me show you this. This is my better image. This is a friend of mine. She's an artist. So this is the animal's preferred activity pattern, right? Sleeping, thinking about food, eating food, right? Hanging out with friends, reproducing. These are things that we want to be doing all the time. However, when there is, say you smell a cat, or you hear rustling, could be a predator. That's when you freeze, right? Because you've learned that those sounds, smells, sights, a predator. That's reaction, subconscious. Then it's pre-encounter, post-encounter, service strike. So that's the service strike part where you're actually being attacked by a predator, and either you probably don't want to keep freezing, right? You probably want to either escape or fight. Those are basically your only options. So by that point, you can't avoid, right? The predator's already seen you and is on you. Avoidance happens before the predator detects you. It's like, I know that if I do this, I will avoid that cat's odor and the cat hurting me at all. So avoidance is this kind of down. You can't be supercharged, stressed out. You've got to be somewhat distanced from the threat, but that'll protect you in the future. So a lot of people think that the word avoidance is bad, right? They're like, ooh, that's always used as a symptom of PTSD. Like everybody's avoiding every situation. But in this sense, avoidance is actually an adaptive response, right? It's actually helping you survive. So does that make sense?
Yeah.
Okay,
Thank
cool.
you.
Well, there's no more questions. So thank you so much, Rob.
Thank you. Thank you, everybody.